Hello, everyone, and welcome to another audio podcast. And this series, the Pathology Storm. This is the Shientist, and I'm going to provide an overview around biomedical visualization and analysis of COVID-19. This is an audio podcast series under the Shientist Chartered Edition. It's a SZtorm Robotics production in collaboration with the Renal Patient Support Group, or the RPSG. And today I'm going to provide, as mentioned, a bit more of an overview around the biomedical visualization of this particular work. And the works, as mentioned in the previous audio podcast, is in keeping with an invited peer review publication that contributed to a scientific book publication. In proceeding around the biomedical visualization and analysis of COVID-19, the coronavirus is divided into four genera. So number one, there's the alpha coronavirus. There's a beta. Number two, there's a beta coronavirus. And three, there is a gamma coronavirus. And four, there's a delta coronavirus. And all of these have in some contexts, all of these different genera of COVID-19 have COVID-19 coronaviruses, I should say, have different sort of taxonomy, and stem from and also along with taxonomy, originate from different species as well. A species from the sort of animal kingdom. So a SARS-CoV-2 virion contains an unsegmented single-stranded positive sense RNA, a genome of around 30 kilobases, which encodes a crucial viral protein and used to create progeny viruses and the four essential structural proteins: cell membrane, membrane, membrane envelope, nucleocapsid, and spike. So as far as airborne viruses are concerned, SARS-CoV-2 is transmitted between humans due to entering of viral particles into the upper respiratory tract. The viral entry occurs by the binding of the spike glycoproteins, which are homotrimers present on the surface of the coronavirus to the ACE2 receptor. So in line with this, the spike proteins are cleaved by host proteases into subunits: S1 subunit for receptor binding and S2 subunit for membrane fusion. Microscopically, the infective cells are forced to translate proteins. So microscopically, the infected cells are forced to translate the proteins required for generating the virus's progeny. So in line with that, the next audio podcast will provide context surrounding the renin-angiotensin-aldosterone system, or the RAS pathway, and the role of ACE2 during COVID-19. This ends the audio podcast on this particular aspect around biomedical visualization and analysis of COVID-19. This has been the Shientist in this series, the Pathology Storm. It's been a Storm Robotics production in collaboration with the Renal Patient Support Group, the RPSG. Thanks for tuning in.